আমাদের আজকের স্পিকার আমাদের সবার প্রিয় সামি ভাই। সামি ভাই বুয়েট সিএসসি 00 ব্যাচের স্টুডেন্ট। এরপরে সামি ভাই ডিপার্টমেন্টে লেকচারার হিসেবে ছিলেন। পরবর্তীতে ইউনিভার্সিটি অফ ইলিনয় আরবানা শ্যাম্পেইন থেকে পিএইচডি করেন কম্পিউটেশনাল বায়োলজিতে। তারপর ইউনিভার্সিটি অফ ক্যালিফোর্নিয়া সান ফ্রান্সিসকোতে পোস্ট ডক্টরাল রিসার্চার হিসেবে ছিলেন। এখন সামি ভাই বেইলর কলেজ অফ মেডিসিনের মলিকুলার ফিজিওলজি এবং বায়োফিজিক্সের অ্যাসিস্ট্যান্ট প্রফেসর। সামি ভাই আর কি আমাদের ব্যাচের তো এখানে অনেকেই আছে, সবারই খুবই প্রিয়। আমার তো সর্ট অফ লাইক এ মেন্টর। তো আমরা খুবই খুশি আজকে সামি ভাইকে পেয়ে। সামি ভাই, তাইলে আপনি শুরু করতে পারেন। থ্যাংক ইউ সামি ভাই আমাদেরকে সময় দেওয়ার জন্য।
থ্যাংক ইউ ভেরি মাচ আতিফ। সো ফর্মালি টকটা শুরু করার আগে আমি আসলে একটু এখানে দুইজন মানুষকে খুবই সিনসিয়ারলি থ্যাংক ইউ বলতে চাই। একজন আবু আসিফ স্যার, স্যারের হয়তো মনে আছে যে আমি প্রথম আসলে আবু আসিফ স্যারের কাছে গিয়েছিলাম, আমি আর নির্জন। আমাদের তখন ইচ্ছা ছিল যে আমরা রিসার্চ করব, আমরা জানতামও না রিসার্চ কি। তা আমাদের প্রথম পেপার দিয়েছিলেন আবু আসিফ স্যার। স্যারের সাথে আমরা কিছুদিন তখন কাজ করি। তারপরে আমার ফর্মালি আমি যদি রিসার্চ কখনো করে থাকি, সেটা আসলে শুরু করেছিলাম সাইদুর রহমান স্যারের সাথে। সো আমি আসলে ভাগ্যবান যে আমি আপনাদের সামনে এখন একটা টক দিতে পারছি, আসলে একটা ইন্ডিপেন্ডেন্ট পজিশনে আসার পরে আলহামদুলিল্লাহ। So thank you, Atif, for the kind introduction. We are going to talk about transcriptional regulation. Give me a second. Okay, so we're going to talk about transcriptional regulation. This is one part of research that our lab spends a lot of time in. This is one slide that I have put just to make everyone on the same vocabulary. So by transcription, we mean give me a second. I'm just trying to set up my laser pointer. Sorry about this. Can you see my laser pointer? No?
না ভাইয়া, এখন লেজার আর রেগুলার দুইটাই দেখতে পাচ্ছি একসাথে, বাট মুভ করে না। লেজার পয়েন্টার মুভ করে না।
This is okay now.
হ্যাঁ, এখন দেখা যাচ্ছে।
Okay, sorry about this. So by transcription, we mean a cell creating mRNAs, these ribbon-like things. I denote mRNA, messenger RNA, using these ribbon-like notations. So a cell creates these mRNAs from the genes. This is a process called transcription, like transcribing the gene into the mRNA molecules. And once we have the mRNA, then the cell creates proteins out of these. And as all of you know, proteins are the functional unit of activity inside a cell. So in that sense, transcription is the functional initial point of all sorts of activities in a cell. Transcription is a highly regulated process, and that is actually our point of interest. And I'll elaborate on that in the next slides. In general, at a given time point of development and in a tissue, we want a gene to transcribe a certain number of mRNAs. If that happens, then we say that it is properly regulated. The transcriptional regulation is going on as we expect. If we see fewer than expected mRNAs, then this is downregulated, which may be an indicator of a problem, like a disease. If we see more than expected mRNAs, that's also a problem. So this is the fundamental question that our lab is interested in. How is transcription regulated? And as I just said, that one point of attraction or appeal of this question is the fact that transcription is very systematic. And what do I mean by that? Here I am showing the transcriptional level So this TPM, this is called transcripts per million. It's a unit of mRNA abundance or count. You can think of it as a count. So we are showing the mRNA counts of 3 genes, POU3F2, NKX2.5, and EEF1A1. The point to note here is in 3 different tissues, cerebral cortex, which is brain, heart, muscle, and spleen, in these 3 different tissues, these genes are very systematically active or inactive. So POU3F2, it is active in brain, not at all at heart or spleen. NKX2.5, just the opposite. It's active in heart and spleen, but please note that in spleen, it is about half as active as it is in, in spleen, it is half as active as it is in heart. And EEF1, again, it is very systematically up or down regulated in all these 3 tissues. And actually, if we look at EEF1, it is in general active in all human tissues, but again, very systematically at different levels. So this is the first question that we want to answer. How do the cells achieve this level of systematic control on transcription? How is gene transcription regulated? And the other appeal that I always need to incorporate in my works, being in a medical school, is changes in transcription levels are associated with diseases. So for example, GDF5 is a gene. If it is downregulated, meaning if mRNA production from GDF5 goes down from the typical mRNA production, like from here to here, this leads to a disease of bone, osteoarthritis, like the thickening of these bone joints. Similarly, if the sonic hedgehog SHH gene, it is upregulated, it can create polydactyly, like this type of extra limbs. And these are very well studied genes. And these are the most visible, I would say, diseases coming out of transcriptional misregulation, loss of control of transcriptional regulation. And technically, transcriptional misregulation is a hallmark of many diseases, including cancer, autism, lots of neuropsychiatric disorders, lots of facial distortions like cleft lip and so on. So this is the disease relevance part of this problem. And in general, from studies of model organisms, we know some stories of the mechanism of transcriptional regulations. So we know that the part of our genome that do not code for genes, also known as the non-coding DNA, the non-coding DNA actually keeps or encodes the information to regulate gene transcription. So what do I mean by that? So here I'm showing the gene, and I just said that the transcriptional level of this gene is very properly regulated, but the responsibility of maintaining that regulation is carried on by the part of the genome that do not code for genes. So the non-coding genome has specific regions called enhancers, like this brown region here. And every gene has at least one or more enhancer associated with it. And the enhancer may not be very close to the gene. It may be very far from the gene. So these enhancers, they are actually hidden within the rest of the, so 2% of our genome, I should put it this way, 2% of our genome encode for the genes. And the rest of the about 97–98% of the genome, they are non-coding. So these enhancers, they are located within these non-coding parts of the genome. We are improving in different types of experiments to identify the locations of the enhancers. And based on studies coming from model organisms, we know that the enhancers, they actually do their job by taking help from other proteins called transcription factors. So transcription factors, they are regulatory proteins. They bind at specific locations or sites within the enhancers. So for this triangle shape, green transcription factor, it binds at its green binding site on the enhancer. This purple shaped this purple colored circle shaped transcription factor, this binds at its specific binding site at the enhancer, and together, when they are bound together, they regulate the transcription of this gene. And this is important that these transcription factors, they are able to bind at their proper locations. The two diseases that I just talked about, they are precise examples of what happens when we do not find that the necessary transcription factors are binding at the necessary locations. So for osteoarthritis, for example, the downregulation of GDF5 happens because a transcription factor called YY1 cannot bind to its binding site because of a mutation in the genome. So these are genetic diseases where the mutations happen in the non-coding part of the genome. So a person, maybe he inherited this mutation in his genome, and because of this mutation, the binding site has changed. So YY1, it can no longer bind to that site, and that eventually leads to osteoarthritis. Similarly, for sonic hedgehog, if ETV, this is another protein, if it cannot bind to its binding site in the enhancer of sonic hedgehog, then eventually it leads to polydactyly. So for most part of this talk, these will be the characters that I'm showing now on the screen. They will be the central characters of our story: transcription factors, enhancers, and of course gene and their transcription. And we are trying to study transcriptional regulation from multiple levels, because as I just said, that transcription factor binding, it is an integral part of the whole process. So the relevant question is, how do transcription factors recognize their target binding sites? And then when transcription factors bind to their binding sites, how do they regulate gene expression or gene transcription? I'm sorry, if in some slides we see the word expression, that technically means transcription. So I try to make everything similar. If in some slides we see expression, it means transcription. So these are two of the most fundamental questions that we have asked since the beginning of my PhD and throughout my postdoc. And what I will talk about today is one recent work where we have shown a new perspective in answering the first question: how do transcription factors recognize their target binding sites? We have shown a relevance of DNA structure in transcription factor binding specificity. So so protein structure is a very well studied area. The structure of DNA in terms of its interaction with different molecular machinery, this is more of a new area of investigation, and that's what I did during my postdoc. And in the second question, how do DNA bound transcription factors regulate gene transcription? I will show one new model that we have developed where we can jointly learn the transcription factor DNA binding specificity, meaning how the different transcription factors identify their binding sites, and transcriptional regulation, a joint modeling approach from a technology called massively parallel reporter assays. I'll describe that when we go to that slide. So these have been my main areas of focus. And right now at Baylor College of Medicine, with our collaborators, we are studying transcriptional regulation at single cell resolution. Again, this is like a very new and emerging area of investigation. I hope that you will find this particular area very exciting, and there might be common interests in answering many questions in single cell transcriptomics and spatial transcriptomics. So the idea is, generally, when we study gene regulation in heart, we do not care about the individual cells. We call it bulk data of gene regulation or gene transcription coming from heart. But with the improvement of technologies, now we can look at individual cell. What are the mRNA counts of each gene in individual cells of heart? So that is the single cell transcriptomics technology. And more recently we have seen like more progress in this technology, in the sense that initially when people started looking at individual cells, they were not able to maintain the spatial location of those cells in the original tissue. I'm sorry, my laser pointer was again stuck. Okay, so it's moving now. But then with more improvement in the technologies, now we can also get the spatial location of each individual cells in the tissue. So ultimately, the data that we're looking at looks like this. It's a data matrix. Every row gives us the information of gene transcription in a cell. So for cell C1, we get what are the mRNA count of gene G1, gene G2, and so on. And also, what is the XY coordinate of this cell in the native tissue. So this is a very powerful technology. And the question that we have like picked up as our first question in this area is, how does a cell spatial location influences its transcriptional regulation, the transcriptional regulation happening in that cell? And I would say that the particular study that I would discuss today, it's not super exciting in the sense that we have just scratched the surface. And what we have seen so far is the spatial organization of cells in the tissue and their communication, intercellular communication. Both of these aspects influence transcriptional regulation. So this is still at an observation level yet, and we are trying to understand how does this happen, and what happens if these organization or intercellular communication are disrupted. Because again, we know that cell-cell interaction is disrupted in many diseases. Organization of cells in a tissue, that's also disrupted in many diseases. So we are trying to correlate, or better to say, causally associate spatial organization of cells, intercellular communication with individual cells transcriptional regulation. So this is kind of an overview of the agenda. I'll try to touch upon three different works that we have done more recently. The manuscripts are either published or available at bioRxiv. So I will try to give a very brief overview of each algorithm and maybe one or two interesting aspects of our findings. And please feel free to interrupt. As I said at the beginning, that I want to like have a good discussion and comments and see if there are opportunities to collaborate on any of these problems. So the role of DNA shape in transcription factor DNA binding. And in general, I think this has been written in the molecular biology texts as if this is a prophecy that transcription factor DNA binding is a sequence-specific mechanism. What do I mean by that? So a transcription factor, it binds its binding site, and people think that, okay, let's take a look at the binding site. If this is ACAG-TCA, then this transcription factor, it always prefers ACAG-TCA or minor variations of it. So people built this kind of models or representations for these transcription factors DNA binding preference, which says that it is like very specific about an A at the first position of its binding site, a C at the second position of its binding site. At the fourth position, it's fine to have a C, but it generally prefers a G because of these differences in length or height of these letters. You can see those different preferences. At the fifth position, this transcription factor mostly prefers to have a T, but it's also fine with a G, and so on. So people understand transcription factor DNA binding as a sequence-specific mechanism. This is like a basic molecular biology textbook, I would say, representation of this phenomenon. And sorry, I'm again stuck. So these models of AC, AGC, TGCA, these models are called the Sequence-Motifs for this transcription factor. And as I just said, that this is studied as a sequence-specific mechanism. However, this is also very well known in the community that these sequence-motifs or the sequence preference cannot explain transcription factor binding in many regions genome-wide. So this same transcription factor, it also binds another location in the genome where the site, where the sequence is completely different from this ACAG-TCA. This is GCTA-TGA. So how is this happening? One hypothesis, one thought that we had, and we didn't make this out of the blue. This was built from very classical studies that talked about the importance of DNA geometry in transcription factor DNA binding. So one thought that we had was the following, that if we look at the DNA structure underlying these sequences, ACA-GTCA and GCTA-TGA, maybe the DNA structure is highly similar. So right now here I'm plotting the value of role, which is a geometric property of DNA. And across these 7 nucleotides, the role values, they look remarkably similar. So our question was, is it the case that the transcription factor is actually recognizing DNA structure, DNA shape? So here is what we mean by DNA shape, and I'll go back to the original question after I spend this one slide on DNA shape. So we write DNA as a linear sequence of ACGT, but of course inside the nucleus it is not a linear sequence. It is a 3-dimensional molecule like this. So here in the 3-dimensional molecule, these yellow tubes, they're representing the backbone of the DNA, and these red and blue, yellow and green, those are indicating the nucleotides. So if red is A, blue is T, if yellow is C, green is G. And as we can see that between these nucleotides in the same base pair, there are both linear displacements, there are angular displacements, and these angular and linear displacements between the bases and between neighboring base pairs that we call shape features. And 4 of these shape features, they have been studied like by structural biologists, particularly people who have tried to crystallize DNA molecules and study their structure for about 30 years now. Helical twist, minor groove width, propeller twist, and roll. These are the 4 more classically studied DNA shape features. And more recently, based on like very powerful computer simulations, people have also generated DNA structural profiles for about 10 more geometric properties. So this is role, as I just mentioned, that if we look at the role values across these 7 nucleotides, it looks like these 2 role profiles, although the sequences are very different, the role profiles, they look remarkably similar. So we hypothesized the existence of a shape motif, and we suggested that protein DNA binding, transcription factor DNA binding, it is a shape-specific mechanism. And the point is, yes, nucleotide sequences, they are determining DNA shape. But the question is, does a transcription factor maintain a bag of words when it is trying to recognize its binding site? Or does the transcription factor recognizing proper structurally, I would say, convenient locations in the genome where it has like proper electron charges that can make its binding with the DNA sufficiently strong? Those are more like molecular biology level questions. I won't get into those, but I can't stop talking about those because we face so much criticism from one part of the community about this whole idea that we needed to actually go in some places against molecular biology textbooks. And but fortunately, we also had a lot of support from like very well established people in the community who think that a transcription factor does not carry a bag of sequences. It is more plausible that it looks at the electron cloud density at this binding site, and those electron cloud densities are determined by the structure of the DNA. So anyway, so then we pursued this idea. We found that most transcription factors, they do have shape motifs, and shape motifs may occur independently from sequence motif. This is kind of an outline of our analysis. We looked at hundreds of transcription factors. We compared our approach against like the well-established algorithms and well-established libraries of sequence motifs, and we still found that about 30% locations of protein DNA binding genome-wide, they do not have the sequence motif, but they do have their shape motifs. So the hypothesis is transcription factors recognize shape patterns in the DNA molecule. And this is an outline of our algorithm. We called it Shape Motif Finder, Shape-MF. It is a classification framework. So, so we start with a set of sequences, the positive sequences that experimentally showed that the transcription factor binds in these sequences. So the experiment can locate a bigger region of DNA sequence. The experiment cannot tell us about the exact location of the binding site. So that is the challenge. Where is the transcription factor binding exactly? And how can we characterize that binding location? So we take a set of positive sequences, experimentally derived sequences where the transcription factor binds. And we take a set of negative sequences. And the negative sequences, it could be like scrambled versions of these sequences. Any of the like statistically acceptable negative controls is fine. And then we apply an algorithm called Gibbs sampling. So using Gibbs sampling, sorry, I missed one step. So once we have these sequences, we transform them into their DNA shape values. So each of these red, green, and blue line curves, they are representing the DNA shape value for one sequence. So red is one sequence, blue is one sequence, but now in the domain of DNA shape. And our question is, can we find overrepresented or more often occurring small patterns of DNA shape within these profiles that would characterize the shape preference for the transcription factor? So we apply an algorithm called Gibbs sampling. This comes from statistical physics, but it also has a lot of applications in optimization algorithms, especially in the Bayesian area. So we apply Gibbs sampling, and we can show that the way we designed it, it satisfies a property called detail balance. But very briefly, we start with a random set of locations in each sequence. And we assume that those random locations are my current guess for the shape preferred binding location of this transcription factor. And then we pick up one sequence. We say that, okay, if this one sequence, if I have a chance to update my binding site location in this one sequence, assuming that my guess for the rest of the sequence is correct, which other window or which other part of this sequence can I pick up? So that is a probabilistic sampling, as I'm trying to show here. And we can talk about the details of the algorithm. But this is a probabilistic sampling. The way, as I just tried to describe, we pick up one sequence at a time. We try to update our guess for the binding site within that sequence, assuming that our guess for the rest of the binding sites in the rest of the sequences is correct. So in that way, once the algorithm converges, we we end up with this kind of small shape windows selected from each sequence. And I'm sorry, my laser pointer. Okay, my apologies about this. I don't know why the laser pointer is stuck again. This Gibbs sampling approach gives us one potential binding site in terms of DNA shape from each sequence. And as you can see that some of these DNA shape profiles coming from this Gibbs sampling approach, they do not look similar to the rest of the profiles, like this one that we are showing at the bottom, this red one. So we go to a second step, which is technically an outlier detection step. And after the end of this second step, we actually eliminate these potential outliers. And we report a shape pattern in terms of range of DNA shape values at each position of the transcription factor's binding site. So what it tells now is this transcription factor at the first position of its binding site, it prefers to have values between 34 and 35 degrees of displacement for a DNA shape feature. This is a cartoon, but let's say this is helical twist values. So at the first position of its binding site, it prefers to have between 34 and 35 degrees of helical twist. At the second position, maybe 33 and 34 point something, and so on. So this is the description of DNA shape motif that that this algorithm finds out. So then we ran this on hundreds of algorithms, and we found that most of the transcription factors, they do have shape motif, and this is not only present in inside data coming from human cell lines, but it is also present in very well controlled synthetic experiments that we call in vitro experiments. And here are some more examples of shape motifs. One interesting aspect of shape motif, and that's what I'll now talk about and finish this part, is it helped us answer several outstanding questions on protein DNA binding. And one of those questions was, if two transcription factors come from the same family, we know that their sequence motifs are remarkably similar. So, but we know that genome-wide, they occupy completely different regions, completely distinct regions. And how is that happening? So when we analyzed those transcription factors in the same family, we analyzed their DNA binding data for their shape motifs. We found that these transcription factors, they actually have very, very different shape motifs. So this is June-B, this is FOS. I'm showing the shape motif for the helical twist geometric property of these transcription factors. And as you can see, so basically their DNA binding site is characterized by TGA, something at the middle, and TCA. So TGA, something at the middle, and TCA. But in terms of shape motif, they're totally different. So this is one mechanistic question that we were able to answer using shape motif. And there are a few other parts, very exciting parts, I would say, in my humble opinion, that we have reported in the manuscript. So this is a published work, and we are still continuing this line of research, incorporating DNA shape information in genomics data. We have recently improved the GIP sampling approach because, as you can probably understand, that the GIP sampling algorithm, it takes a long time to converge. So we have recently designed a convolutional neural network approach to study this problem, which I'll touch upon in the next part of my talk. So this is where I would wrap up on the first part on the role of DNA shape in transcription factor DNA binding and specificity. In the second part of our talk, I'm going to talk about a model where we try to answer that second question. If two transcription factors are binding to an enhancer, how do they regulate the transcription of the target gene of this enhancer? So in this part of the talk, we have one new player in the story. This is the basal transcription machinery, this star-shaped thing. So basically, these transcription factors, they help to recruit another protein or a protein complex. Altogether, we call them the basal transcription machinery at the gene promoter, at the starting position of this gene. And once the basal transcription machinery is recruited by these transcription factors, the gene is transcribed. The more frequently these collection of transcription factors are capable to recruit the BTM, the more frequently this gene will be transcribed. So we studied this problem throughout my PhD. The framework that we used to study the problem was a classical framework proposed by Shia and Akers. So what they said is, imagine this system of these three different players, TF1, transcription factor 1, transcription factor 2, and BTM. This system can be in any possible configuration out of these 8 configurations. So configuration 1, we have only the BTM, but it's there just by a random chance, not recruited by the transcription factor. Configuration 2, it could be that the second transcription factor is here and it has recruited this BTM. Configuration 3, again, only the green transcription factor is here and it has recruited the BTM. So in this way, we can imagine 2 to the power 3 number of possible configurations. So Shia and Acker said that if this system, if it is possible for this system to stay in any of these 8 configurations, what is the equilibrium probability for this system? And the equilibrium probability for the system would give us the expectation of the configurations where I have the basal transcriptional machinery sitting here. And those probabilities taken together would be proportional to the gene transcription level. So this was their more theoretical framework. And we realized that we can model this whole thing as a probabilistic graphical model. Each of these configurations now become a state in the model. And there is the transition probability from one state to another state. And that transition probability, it represents the physical property of this transcription factor to dissociate or to bind at its binding site. So now we are talking about a probabilistic graphical model, and we are talking about a subset of that, of the state space of that graphical model, where we have this BTM bound at the promoter. And then we can just take the expectation of those probabilities, and the observed gene expression data would be the expected value of those configurations where I do have this BTM bound. And my apologies, this should be 4 here, because the observed gene expression data, as I just described, the Shia and Acker's framework suggests that the observed gene expression, the data that we are trying to model, is the expected value of those configurations where I do have the BTM at its promoter. So this should be 4, my apologies. So as I just said, that this can be modeled efficiently as a probabilistic graphical model. Although we are talking about exponential number of states in the model, we can think of a dynamic programming algorithm to efficiently calculate this expectation term. The point of this model is we need to know the transcription factor DNA binding preference for every possible transcription factor, and that may not hold. That may hold in yeast, that may hold in fruit fly, in other model organisms, but that may not hold for human genomics data. So we may not know the protein DNA binding specificity for all the human transcription factors. So a little bit of informative piece here. As I showed before, that the protein DNA binding specificities, they are modeled using this kind of logos, where it actually shows that what is the preference for this transcription factor at different positions of its binding site. If it mostly prefers, like 97% of the time, if it prefers to find an A, we will put a like tall A at the first position. If it is like 95% of the times, if it is G, then we will put a tall G here. However, if it is like equally likely to have a C or a G, so this G and C, they are equally likely, and in terms of information content bit, this transcription factor is not actually very specific about any particular nucleotide at this position. So this information content value goes 0. I think it's much easier to imagine the binding specificity model as these probability matrices rather than the information content based representations. But this is intuitive, as we can see that if a transcription factor is very much biased to a particular nucleotide, we see that in this representation, we see that that character, that letter nucleotide, is drawn as a tall character. That's the basic model. So how do we model this enhancer to gene transcription data sets if we do not know the binding specificity of these transcription factors? So one approach could be to learn the transcription factor DNA binding specificities right from the data, and there we can design expectation maximization algorithms for those, but again, those are like difficult to train. So more recently, people, most of the genomics labs, we have moved toward like convolutional neural networks, and the idea is using the convolutions Can we learn the transcription factor DNA binding specificity? So in general, the CNN, the convolutional neural network models in this realm look like this. We encode the input enhancer sequence as an one hot encoded vector. We use a bunch of convolutions. Each convolution is a matrix, and each convolution tells us some number that can be positive, that can be negative, that can be zero, telling us what are the preferred nucleotides at each position. So each of these convolutions in that sense represents this kind of a probability matrix with the caveat that the convolutions, they are not really learning the probabilities. They are just learning, as I said, some numbers. And we don't know how the neural network model is integrating those numbers. So typically, as we know that we use convolutions, we use some form of pooling, and this whole block repeats for 2 or 3 more times, then we have a fully connected layer and so on. The point is, these convolutional neural networks, once we try to interpret those, once we want to understand what's happening at the convolution level, can I interpret the convolution as this kind of a probabilistic matrix, which also has strong biophysical meanings? We fail. We need post hoc analysis to identify what the convolution is actually doing. And even though there are very good algorithms to identify what this convolution is trying to model, the point is that is a post hoc analysis kind of detached from the model itself. It is not guaranteed that the same convolutions that I now get from the post hoc analysis will provide an equally good model. This is trying to understand what the model is doing, making some more assumptions on the model's behavior itself. And finally, interpreting what's happening at the convolution layer, at the first layer, does not mean that we have identified a biophysical mechanism at the downstream part of the model. So we started thinking that how about if we can impose some constraints on the convolution themselves? In other words, can we learn the convolutions as this type of probability distribution? So here, each column is a probability distribution. Each column is telling us about a multinomial distribution. So we asked, is it possible? Would it work if we use each convolution to actually fit a multinomial distribution? And it turned out that it is really easy, and it was remarkably effective. What we did was, so each convolution, it is this X matrix here. So we allow each convolution still to learn all sorts of values, 0, minus, negative values, positive values, everything. But before we apply the convolution on the input sequence, before we perform the convolution operation, we transform these values into a multinomial distribution. So as you can see, this is also known as the Boltzmann transformation. We exponentiate each value of the convolution matrix, and now that takes it to a positive value, a non-negative value, I should say, and normalize those values with the summation of those exponentiated terms across every row. So now every row, it gives us 4 of these terms which sum up to 1. Each of those are non-negative. So now we can interpret every value as a probability value. Now these convolutions, they become multinomial distributions. And they can help us create very easy to interpret, biophysically interpretable models. Using these multinomial distributions, we can compute likelihood terms. They go into very simple linear regressions. Overall, this is the model that we found to be as effective as the very complicated models we have seen in the literature. We learn every convolution as a multinomial distribution. We take the max pooling values, like these convolutions now can tell us, according to this probability distribution, what is the most likely location where this particular transcription factor will bind in this sequence, and we apply a simple linear regression. We fit the gene expression. So this was a very simple model, and yes, we found good things about the model. As I just said, that this is simple, easy to integrate. We do not easy to interpret. I'm sorry. I'm sorry if I'm rushing. Easy to interpret. We do not utilize any information beyond the sequence information, and it improves over the state-of-the-art performance. So this is deposited in bioRxiv. If you are interested, you can take a look at the model, and I will just show one interesting application of the model on unseen data. So now we learned this model from a bunch of enhancers, and we asked, is this model of any good to us? So we took another data set where about 200,000 sequences were scored according to their ability to act as enhancers. So we asked, okay, if I take these 200,000 sequences, and if we want to rank them according to our model, how strong will those enhancers be? Then how does our model rank those sequences? And this is how our model ranks these sequences according to the predicted activity of these sequences. And this is the rank. And as you can see, that the predicted activity of these sequences correlates very strongly with the rank, which was like remarkable. At least to us, this was remarkable, because the model did not learn these data during training. The model trained itself on a different set of data, and this is a different type of enhancer activity data. So on a totally unseen data, we found that the model is showing us very credible predictions of enhancer activity. So then we asked, okay, if I now try to interpret whether a mutation in the genome is good or bad, can the model do that? Can the model show that if we have a mutation in an enhancer, that mutation creates a huge change in gene expression? So potentially that is a bad mutation. So again, we looked at public databases of human genome mutations, and we said that let the model find which mutations have the highest predicted value of expression change. So we call that predicted variant effect. And the model actually found about 200 mutations, which give us significantly altered gene expression value. Now, how do I believe whether those 200 mutations can do something bad? Technically we believe that, because if we look at the frequency of those mutations in the human population, those mutations are all very rare mutations. So this is minor allele frequency, a term to indicate how common is a mutation in the human population. And all these strong gene expression activity changing mutations, they're very rare in the human population, which comes along the understanding that if a mutation is bad, we won't see that as a common mutation in the population. That would be suppressed in the human population. So I would wrap up the second part of the talk here. Again, this model we just published. I wouldn't say published. We just deposited the manuscript in bioRxiv. I welcome more thoughts and comments on this. But as I said, that I think this is an interesting way to approach building interpretable convolutional neural networks. Can we embed individual distributions into the convolutions so that we can readily interpret the convolutions? Now, going back to our shape motif idea, now we are trying to implement convolutional neural networks to discover shape motifs, but the convolutions, they are trying to learn Gaussian distributions. Very simple, one Gaussian distribution per position. And again, we are seeing that this is working. So I think this is an interesting direction, again, at least for us, that we have struggled, at least the genomics and the biomedical community have struggled for quite some time to interpret these models, because it's not a YouTube recommendation. This is like a diagnosis about an individual's health. So we need to understand what the model is doing. And this is one important step toward understanding what the model is doing. Can we embed the distributions within the convolution? So the convolutions will learn the distributions, and then we'll just take the convolutions, look at them, rather than doing all those complicated post hoc analysis. So in the last part of my talk, I'm going to touch upon our recent work on spatial transcriptomics and single cell transcriptomics. So this is the schematic of the whole idea. We are looking at mouse brain. So this left panel, this is showing a cartoon of a mouse brain. We take a slice. It's not us, I'm sorry. the experimental biologists, they take a slice of the mouse brain and then they put it, let's say, under microscope. So this is how this slice looks like when put under the microscope. And these different lines in this part of the brain, they just denote different layers of different types of neurons in the mouse brain. So if we do a single cell experiment on this sample, then the single cell experiment would bring this type of a data matrix for us. Every row is a cell, every column is a gene, and for every cell we get the mRNA count for that gene inside that cell. If we do a spatial transcriptomics experiment using the same sample, we would get this type of a data matrix where with each cell we would also get the XY coordinate for that cell inside this tissue sample. But it is important to note that spatial transcriptomics, because of limitations of the technologies, they can profile much fewer genes. So I put it here on the favor of single cell, that they can profile many more genes. So as I showed here, that maybe the spatial transcriptomics, it is not capturing the expression of gene G2 in this tissue. It has gene G1, it has G3. In general, the value of K is about half as many, is about half as much as the value of N. So single cell brings a lot of information about gene expression, but single cell does not bring information about XY coordinate, which spatial transcriptomics does. And also, now we are talking about two different samples. So if I am getting data from cell C1 here in single cell, it's a different cell that I'm collecting data under the spatial transcriptomic experiment. So that's why I made them, and that's why I put them under different notations. In other words, more visually, this is how we tend to think about the data. So single cell, it brings information about a lot of genes and a lot of cells. And here, so it's more like a scatter plot. Each dot represents an individual cell. And an individual cell, it is characterized by the expression of about 20,000 genes. So to visualize the cells on a two-dimensional plot, we need to apply some dimensionality reduction method, which is very well established. If we apply those dimensionality reduction methods on this single cell RNA-seq data matrix, it reduces each cell to two informative coordinates, and then we plot each cell along those coordinates, and we see that very similar cells, they are clustered together in this low dimensional representation. So each color represents one type of neuron, as we can see, or one type of microglia. So this was a breakthrough finding from the single cell studies, that we talk about brain, but brain actually comprises many different types of cells. And understanding these individual types of cells is very critical for disease diagnosis, understanding and designing therapies, and so on. So single cell RNA-seq brings us very precise information about these different cell types, because single cell RNA-seq tells us very precise featureization of each individual cell. On the other hand, spatial transcriptomics, it tells us about XY coordinate, but it just brings the cell. And as I said, about half as many genes as single cell RNA-seq does. But based on those fewer number of genes, it's difficult to assign cell type information to each cell in a spatial transcriptomics assay. Ideally, we want to have this kind of color in both assays. But in spatial transcriptomics, we do not get the colors. We do not know what is the type of the cell, because we get information about much fewer number of genes. So then maybe we thought that maybe we can try to get a model which will learn cell types from the single cell data and map the cell type information on the spatial transcriptomics data. And again, the caveat is the model has to work on only those genes that are common between single cell RNA-seq and spatial transcriptomics. So we are talking about only the shared genes between the two technologies, and it may not always be possible to do that. So in our manuscript, we tried to present a sufficient condition which can at least check whether this is doable. So for this data set, this was doable. We ended up finding these colors, these cell types for each individual cell in the spatial transcriptomics data. So for now, for this mouse brain sample, we know the XY coordinate and the type of each cell. And each cell also tells us about the gene transcription data inside that cell. So then we asked, okay, how are transcriptional activity changing from one cell to another cell? So this data set, it actually collects data not from the entire mouse brain. It collects data from small sections of the mouse brain. So these are the 7 small sections in the mouse brain from which we got data. So each small rectangle here tells us one region from which we got spatial transcriptomics data. So when we analyzed the data and we mapped the type of each cell in each of these small rectangular areas. So for now, I'm looking at this rectangular area annotated with 2. I'm zooming in, and each of these boxes, it is showing us the location of one type of cell. So in this case, this is showing us the location of neuron transition cells in box 2. Here, it is showing us the location of olfactory ensheathing cells in box 2. So these are not exact locations. These are kernel density estimates, because exact locations, they're pretty sparse. So we ended up using kernel density estimates. What is remarkable here is if we compare what's happening in terms of organization on the space for neuron transition cells and olfactory ensheathing cells in this box 2 and box 3, as we can see that neuron transition and olfactory ensheathing, they're pretty much exclusive to each other in box 2. And they are nearly overlapping with each other in box 3. So this is, I guess, more easy to visualize here. So, I mean, maybe this is also clear from here that these dark regions for olfactory ensheathing, they fall at the low abundance regions for neuron transitions. So that's what we see here, that these two cell types, they're pretty much disjoint. Only these common hatched areas are where they co-occur, but they happen most of the times disjoint, exclusive to each other. However, in this box 3, they are super overlapping with each other, as we can see here. So this was our first like I would say piece of information that we gained, that even within a tissue and even within a very like uniform morphological layer, we call these morphological layers because within these annotated regions, experts say that the function of the brain remains very similar. So the point is, even within this part where everything is functionally similar, we see this much variation in co-localization of different cell types. So then we asked, okay, if two different cell types are co-localized, or if they're staying away from each other, how does that alter their intercellular communication behavior? And there are established tools to study cell-to-cell communication. We used those tools to ask which receptors and which ligands, which proteins do these neuron transition and olfactory ensheathing cells use with each other to talk to each other. And we actually found that there are remarkable variations in the use of receptor ligands across the space. So in general, we find that from one part of the tissue to another part of the tissue, there can be 15-fold change in the number of receptors and ligands that two cell types are using to talk to each other. And more importantly, if we look at those receptors and ligands, we see that those receptors and ligands in different part of the tissue, they are activating different cells, they're activating different genes, or they're being activated by different genes across the space. So now we are looking at huge amount of heterogeneity, huge amount of variations in the mechanism of transcription regulation across spatial locations in a tissue. So as I said at the beginning, that this study was more of a scratching the surface. It just told us And we were able to quantify in many aspects how spatial organization of cells and intercellular communication influence transcriptional regulation within individual cells at their native location. And now we are going after more targeted hypothesis to understand how this is happening and how these are changed in a diseased context. So I think I would start wrapping up here. To summarize, we discussed 3 parts of our recent attempts in answering mechanisms of transcription regulation. One is the role of DNA shape. We published this very recently in Cell Systems. The second is jointly modeling transcription factor DNA binding by using a probabilistic convolutional neural network. We put that in bioRxiv more recently. And then spatial transcriptomic and single cell analysis showed us that spatial organization and intercellular communication, they influence transcription regulation remarkably. So this is also in bioRxiv. Before I finish, I would like to acknowledge my postdoc advisors, Katie Pollard and Benoit Bruniou. Peter von Hippel and Ron Schmidt, they're very famous names in the area of transcription factor DNA binding. They were very encouraging on our idea of DNA shape motifs. Funding sources and collaborators at Baylor College of Medicine, Jim Martin and Diwakar, we are working with them on single cell and spatial transcriptomics of human heart, developing human heart and disease human heart from pediatric samples. Susan, we are working with her to understand the genomics of metabolism. And with Robia, we are working to understand the genomics of Alzheimer's disease. And this is my lab right now. It's a very small lab. And as you can imagine that these are remarkably energetic people who are taking on all these collaborations. So Salvi, she's looking at DNA shape and the idea of probabilistic convolutions. Minjun, she is looking at single cell data. Francisco, he's looking at single cell and spatial transcriptomics. Jian, I didn't present his work today. Jian is looking at mutation patterns and do DNA structure have any role in the mutation patterns that we typically see in healthy population or in disease population. And finally, even if you are not asking any question today, or if you are not expressing your annoyance today, please feel free to send your comments or suggestions to my email. It's samee@bcm.edu. So I think with that, I can wrap up, and I'm happy to take any questions and suggestions. Thank you for the privilege of your time.
Thank you, Samee bhai, for a wonderful talk. আমরা question answer এ চলে যাই। question থাকলে either hand raise করলে হবে অথবা চ্যাটে টাইপ করতে পারেন। হ্যান্ড রেজ করে তারপরে আমরা একে একে আনমিউট করে তাহলে কোশ্চেন করতে পারবেন। হ্যাঁ, আশিস রাহা।
সামি ভাই, শেপ মোটিভ সম্বন্ধে আরেকটু বলবেন যে শেপ মোটিভ আর সিকোয়েন্স মোটিভ কতখানি একে অপরকে রিপ্রেজেন্ট করে, যেসব সিকোয়েন্স মোটিভ আছে তার কত পার্সেন্ট আপনারা শেপ মোটিভের মাধ্যমে রিকগনাইজ করতে পারেন অথবা ভাইস ভার্সা?
So in general, shape motifs, they actually complement sequence motifs. They cannot replace them. As I said, that sequence, it determines DNA shape. So to answer your question, shape motifs grab all the sequence motifs. That's almost always true. However, shape motifs, they grab additional nucleotides flanking the sequence motifs. So, and sometimes shape motifs, they also explain additional sequences which look very different from sequence motifs. So the short answer is shape motifs, they are just more comprehensive. They grab the sequence motifs plus more because of the similarity of DNA shape. Does that answer your question, Ashish?
হ্যাঁ, ঠিক আছে। আমি জাস্ট আরেকটা শর্ট কোশ্চেন, সেটা হচ্ছে এটা কি ট্রান্সক্রিপশন ফ্যাক্টর তো প্রমোটার সাইডেও বাইন্ড করতে পারে, রাইট?
রাইট।
আপনি কি আপনার এই মডেল কি শুধুই এনহ্যান্সার সাইটে বাইন্ডিং রিলেটেড, নাকি এটা এনহ্যান্সার প্রমোটার ইররেসপেক্টিভ কাজ করতে পারে?
Enhancer promoter irrespective. I mean, the model does not make any assumption about a particular class of the sequence. just like any DNA sequence, as if it is coming from a synthetic sequence library. Even that is fine.
আচ্ছা, আরেকটা হচ্ছে আপনি এখানে মাঝখানে যখন কোড করছেন জাস্ট দু একটা স্লাইড পিছনে, সেখানে হচ্ছে না, যেখানে হচ্ছে আপনার শেপ ভ্যালু কম্পিউট করছে, আমি ঠিক ক্লিয়ার ছিলাম না শেপ ভ্যালুটা কিভাবে কম্পিউট করছেন।
Give me a second.
হোয়াটসঅ্যাপ স্লাইড স্যার, যদিও স্লাইডের সংখ্যা কম কিন্তু অ্যানিমেশনের অনেক।
I don't know if that is a compliment. Okay, so I think I tried to move very fast along this from in this part of the talk. So this part, we have these sequences where we're getting the DNA shape value of each sequences. So now the question is, what are the small patterns of DNA shape that are very similar across different sequences, right? So as if
ওই জায়গাটাতে আপনি সিকোয়েন্স থেকে কিভাবে শেপ ভ্যালুটা পান, বা আমি জাস্ট
so these are coming from another set of tools which predict the DNA shape values at each nucleotide position. Those tools, they're made from simulation algorithms, and they have been shown to be very, very accurate these days by showing their accuracy with respect to crystal structures and NMR structures. So that's why people actually do not question the quality of the DNA shape data. They question our hypothesis.
ঠিক আছে, ঠিক আছে।
We were told at Berkeley that we are flying against the face of molecular biology.
আচ্ছা, আমার আরো কিছু কোশ্চেন আছে, বাট আমি সব টাইম নিচ্ছি না, আমি অন্যদের সুযোগ দিচ্ছি, পরে কোশ্চেন করব।
Sure.
নেক্সট আর কারো কোনো কোশ্চেন?
আমি এই শেপের বিষয়ে একটা কোশ্চেন করতে যাচ্ছি সামি, সেটা হচ্ছে শেপ ফিচার, মানে যেই ছবিটা দেখা যাচ্ছে একটা সিঙ্গেল লাইন, যেটা হচ্ছে তার কিছু অ্যামপ্লিটিউড আছে বেসিক্যালি, মানে এটা কি যে আগে যেটা দেখালো যে 4 ধরনের স্ট্রাকচার হতে পারে, সেগুলোর মধ্যে যে কোনো একটা পার্টিকুলার স্ট্রাকচারের জন্য তার অ্যামপ্লিটিউড ভ্যালু, নাকি এটা একটু বোঝা দিলে সুবিধা হতো?
জি ভাই, এটা আসলে যে কোনো একটা স্ট্রাকচারের জন্য। সো আমরা আসলে শেপ মোটিভ লার্ন করি ওয়ান ফিচার এট এ টাইম, তারপরে আমরা স্টাডি করি whether the shape motifs or different features they coincide or they kind of explain different parts of the binding site independently. কিন্তু initially আপনি যেটা বললেন ভাই, initially the model runs on one shape feature at a time.
আচ্ছা, আর এরপরে এই স্লাইডের ডান দিকের পিকচারে যেই মানে একটা রেঞ্জ মতো শেড করে দেখানো হয়েছে, সেটার মানেটা কি যে যেগুলো ক্যান্ডিডেট হয়েছে তাদের সর্বনিম্ন থেকে সর্বোচ্চ পর্যন্ত কত ছিল সেই ভ্যালুগুলা?
জি ভাই, এটা মোর লাইক আ উইগল রুম আপনি যেটা বললেন। তবে আমরা যেটা দেখি আসলে যে এটা আসলে এই স্টাডিতে আমরা আসলে রেঞ্জ হিসেবে মডেল করি, কিন্তু আসলে এটা গাউসিয়ান ডিস্ট্রিবিউটেড। সো দ্যাটস ওয়াই আমরা ওই যে পরে প্রবাবিলিস্টিক কনভলিউশনে আমরা আসলে এখন এই প্রতিটা পজিশনকে এক একটা গাউসিয়ান হিসেবে ফিট করতেছি।
আচ্ছা, আরেকটা কোশ্চেন রিলেটিভলি যদি জিজ্ঞেস করি, সেটা হচ্ছে যে এখানে যেমন শুরুর দিকে যেরকম বোঝাচ্ছিল, তাতে আমার কাছে মনে হচ্ছিল যে মানে এখানে কতটুকু বেশি, যেমন ওই ছবি না, সরি ওই ছবিটাতে যদি আসো, বা এই ছবিটা তো ঠিক আছে, এই স্লাইডের ছবিটাতে আপাতত যদি থাকো, যে জাস্ট দ্য ওয়ে এখানে যেটা দেখা যাচ্ছে, মানে এখানে আইডিয়াটা আমার কাছে মনে হচ্ছে অ্যাজ আই গো ফ্রম লেফট টু রাইট পজিশনে, তাতে আমার একটা আপওয়ার্ড বেন্ড আছে, তারপরে ডাউনওয়ার্ড বেন্ড, দেন আপওয়ার্ড, ডাউনওয়ার্ড এন্ড সো অন। ওই ডান দিকের ছবিটা তো তা আছে, মেবি একটু কম আর বেশি এক্সটেন্টে, রাইট? এখানে কি মোর ইম্পর্টেন্ট দ্য টার্মস, মানে দ্য রিলেটিভ অর্ডারিং গুলাই, নাকি এখানে ওই অ্যামপ্লিটিউড গুলো তখন ইম্পর্টেন্ট হচ্ছে?
বোথ ভাইয়া, বোথ। আসলে আমরা তো আলটিমেটলি এটাকে একটা ক্লাসিফিকেশন অ্যালগরিদম হিসেবে সেট করি। সো দ্য ডিসক্রিমিনেটিভ ফিচার বিটুইন পজিটিভ এন্ড নেগেটিভ, ওই ডিসক্রিমিনেটিভ ফিচার হিসেবে দেখা যায় যে অ্যামপ্লিটিউডটা ইম্পর্টেন্ট, এন্ড দেন দ্য চেঞ্জেস ফ্রম ওয়ান পজিশন টু অ্যানাদার পজিশন, যেটা আপনি ভাই বললেন যে টার্ন, সেটাও ইম্পর্টেন্ট আসলে।
আমি যে জন্য জিজ্ঞেস করছিলাম সেটা একটা হচ্ছে যে অর্ডার প্রিজার্ভিং প্যাটার্ন ম্যাচিং বলে একটা জিনিস আছে, যেখানে আসলে ওই যে মানে পার্টিকুলার ভ্যালু না, কিন্তু স্ট্রিং এর মানে একটা সিকোয়েন্স অফ নাম্বারস এর মধ্যে কোনো আমি যদি একটা পার্টিকুলার প্যাটার্ন বলে দিই যে এই প্যাটার্নে চেঞ্জ হচ্ছে, এখন সেই জিনিসটা আমি লিনিয়ার টাইমে সাম আদার আর কোন কোন জায়গায় অকারেন্স করছে এটা লিনিয়ার টাইমে কম্পিউট করা যায়। তো ওই রকম কিছু এখানে কাজে লাগবে কিনা সেটা একটা আমার কিউরিওসিটি ছিল।
আমার মনে হয় ভাই অবশ্যই কাজে লাগতে পারে। আমি আসলে ওই প্রবলেমটার কথা জানতাম না, আপনি যেটা বললেন।
আমি তোমাকে একটা পেপার ফরওয়ার্ড করব না।
জি, ইন জেনারেল আমরা আসলে ইনিশিয়ালি ভাবতাম যেটা আসলে টাইম সিরিজ মোটিফের সাথে এটা রিলেটেড, কিন্তু পরে আমরা যেটা দেখলাম যে টাইম সিরিজ মোটিফের এখন সবচেয়ে ভালো অ্যালগরিদম যেগুলো মানে রিভার্স সাইডের ইমন কিউ, ওরা যে কাজগুলো করে ওরা আসলে প্রবলেমটাকে টোটালি অন্যভাবে ডিফাইন করে। সো টাইম সিরিজ মোটিফের সাথে আসলে এটা তখন আর টাইম সিরিজ মোটিফের অ্যালগরিদম গুলো এখানে তখন আর আমরা আনতে পারি না। তো ওই জন্য আসলে আমাদের কাইন্ড অফ গিফট স্যাম্পলিং দিয়ে কাজটা শুরু করতে হয়েছিল।
সামি ওই যে হ্যালো, হ্যাঁ সামি ওই যে তোমার যে চারটা ফিগার যে জিওমেট্রিক ওইটার ওই স্লাইডটা একটু ওখানে।
জি স্যার।
আচ্ছা, এখানে মানে যেটা আমি দেখতে পাচ্ছি এখানে তোমার এই যে মানে রোটেশনাল, তারপরে এটি, এবং এখানে যদি তুমি রিজিড স্ট্রাকচারের কথা চিন্তা করো তো রিজিড স্ট্রাকচারের ক্ষেত্রে তার ডিগ্রি অফ ফ্রিডম, তারপরে এই সমস্ত জিনিসগুলো কিন্তু এখানে চলে আসে কিছু জিনিসপত্র। মানে রিজিডিটি থিওরি এখানে অনেকেই ব্যবহার করেছে, তুমি এগুলো কি দেখেছো এই জিনিসগুলো?
না স্যার, আমি আসলে ওগুলো কনসিডার করা হয় নাই, বাট আমরা এখন স্যার যেহেতু আরো ফিজিক্যালি জিনিসটাকে বুঝতে চাচ্ছি, আমার মনে হয় এই ইনপুটগুলো আমার জন্য খুব হেল্পফুল হবে স্যার। আমি নোট রাখতেছি স্যার। স্যার রিজিডিটি থিওরি বললেন স্যার?
হ্যাঁ রিজিডিটি থিওরি মানে কম্বিনেটোরিয়াল রিজিডিটি ইয়েতে।
আচ্ছা স্যার, ইন জেনারেল স্যার। না স্যার আপনি বলেন, না আপনি বলেন প্লিজ স্যার।
মানে ওখানে তোমার ওই রিজিডিটি থিওরি দিয়ে ধরো মানে একটা রিজিড স্ট্রাকচার কিভাবে ডিফাইন করে, এবং এই জায়গায় তোমার ফাংশনাল ডিপেন্ডেন্সি এবং রিজিডিটির সাথে বেশ একটা ভালো রিলেশনশিপ আছে। কিছু রিসার্চার এগুলো মানে কাজ করেছে আর কি গত কয়েক বছরে, তুমি একটু দেখতে পারো আর কি।
ডেফিনেটলি স্যার, ডেফিনেটলি। আমি আসলে স্যার যেটা বলতে চাচ্ছিলাম যে প্রথমে যখন আমরা কাজটা শুরু করি তখন মানে লাইক এনি আদার প্রজেক্ট, প্রথম কথা ছিল যে ইজ দেয়ার আ সিগন্যাল। এখন যখন সিগন্যাল দেখলাম তখন পয়েন্ট হচ্ছে যে এটাকে আমরা কিভাবে এক্সপ্লেইন করতে পারি। সো এখন স্যার আসলে এটা আমাদের রেসপন্সিবিলিটি টু ট্রাই টু এক্সপ্লেইন ইট। বাট আপনি যেটা বললেন স্যার আমি মনে করি এটা ইম্পর্টেন্ট, কেননা আমাদের কিছু কোলাবোরেটর স্যার বলে যে একটা টিস্যুতে যখন একটা টিউমারে যখন হচ্ছে গিয়ে ট্রান্সক্রিপশনটা রেগুলেশনটা নষ্ট হয়, ওরা মনে করে যে টিউমারের যে ভলিউমের তুলনায় সেল অনেক বেশি, সেলগুলো একটা আরেকটার উপরে মেকানিক্যাল স্ট্রেস দেয়, এবং সেই মেকানিক্যাল স্ট্রেসের জন্য ডিএনএ স্ট্রাকচার চেঞ্জ হয়। আপনার সাজেশনটা স্যার প্রবলি আসলে ওই লাইনের থিংকিং এ আমাদেরকে হেল্প করবে।
হ্যাঁ ওই ইয়েতে মানে যেমন ধরো ওই যে রিজিডিটি একদম সিম্পলেস্ট ওয়ে বলা যায়, ধরো একটা যদি মানে চারটা দিয়ে একটা চতুর্ভুজ বানালা চারটা কাঠি দিয়ে।
জি স্যার।
এই স্ট্রাকচারটা তুমি চারটা জয়েন্টে যতই তুমি রিজিড ভাবে আটকাও না কেন, স্ট্রাকচারটা কখনোই রিজিড হবে না। তুমি এটার উপরে যদি স্ট্রেস করো এটা মানে হয়ে যাবে আর কি, ডিফর্ম হয়ে যাবে। তুমি চারটা জয়েন্ট যতই তুমি ওখানে শক্ত করো না কেন, কিন্তু মাঝখানে যখন তুমি একটা ট্রায়াঙ্গেল দিয়ে দিচ্ছ আর কি ইয়েতে, মানে একটা ডায়াগোনাল দিয়ে দিচ্ছ, তখন কিন্তু ওইটা একেবারে রিজিড হয়ে গেল। এটা হলো একদম বেসিক থিং আর কি রিজিডিটি থিওরি। হ্যাঁ, এইটার হচ্ছে এই যে স্ট্রাকচারাল, এইটাকে যদি আরো বড় স্ট্রাকচার হয় এবং কিভাবে, এবং এটা দিয়ে আর কি এই জিনিসগুলোর ফাংশনালিটি এক্সপ্লেইন অনেকেই করার চেষ্টা করছেন আর কি এই থিওরির সাথে। বেশ কিছু থিওরিও আছে এই রিলেটেড।
স্যার আমি আপনার সাথে যোগাযোগ করি স্যার, যদি আর কি আপনি আমাকে এরকম কয়েকটা পয়েন্টার দিতেন, মানে সাইফুর ভাই আপনি মানে আপনারা একটু পয়েন্টার দিলে আমার মনে হয় আমাদের জন্য এটা খুব ভালো হবে, মানে জিনিসটাকে আমরা আরো তখন ফিজিক্যালি রিপ্রেজেন্ট করতে পারবো।
ঠিক আছে।
জি স্যার।
হ্যাঁ, সজন মাহবুব।
আসসালামু আলাইকুম। Thank you very much for your wonderful presentation. I had a question about your second project. Could you please go to the slide where you talked about the convolutional neural network?
Yeah, this one.
Yes, yes, thank you. So are you trying to predict whether a transcription factor will bind to a non-coding part of the DNA, or just what to say?
Okay, so the answer is yes. The way I presented it, this was based on a data set which is more appropriate for a regression modeling. But yes, I think even if I come to your question, let's say I have the same data set of transcription factor binding, a set of sequences where the transcription factor binds, and a set of control sequences where it does not bind. And I'm trying to predict what are the most overrepresented patterns of sequences in the binding sites, in the bound sequences, that distinguish them from the control sequences. So to make that prediction, yes, we are using this convolution.
Thank you. So I had another question regarding this. Are we also trying to predict where in that sequence the transcriptor factor is actually binding, or we already know that information from the data set?
Exactly. We have to predict that because the experiments, they actually pull out a longer sequence, like 100 bases, and the transcription factor binds somewhere within that 100 bases, and it takes like 10 to 15 bases for binding. So that's why we need to find overrepresented patterns from those longer sequences. So you are right. We are trying to predict the patterns that the transcription factor prefers, and where in the bigger sequences do those patterns happen.
Thank you. Basically, I did a sequence-based work, something similar to this, but that's based on speech data, where we try to figure out for a sequence where we just predicted, we just had the label for the whole sequence, whether there is whether there is a smile or not, similar to this, whether it will bind or not, and we used multi-instance learning, which gives us a probability distribution and tells us whether whether it binds or not, and at the same time tells us which are the places this may bind, which is not explicitly given in the dataset as a label, but the model gives us a probability over that.
And the probability distribution is over what? I mean, what is the random variable for that distribution?
So the probability distribution is over the input sequence. So in this case, the individual base pairs. So that will give us a probability distribution over the base pairs, basically.
I see. So here is one interesting point about DNA sequences. Technically speaking, we already know that if there is an experiment done properly, then the transcription factor binding site, its spatial location at a base pair resolution would be pretty much Gaussian distributed around the center of that binding of that sequence. So that's why we already clipped the sequence to 100 bases or 150 bases around the maximum possible probability for that transcription factor to bind. But it is still a huge variance, like 150 or 100 bases. So then we have to come up with these models, which A, define the preference of the transcription factor, and B, also can tell us what is the most likely position for this transcription factor to bind within these 100 bases. But I think there are interesting similarities, but also some unique characteristics in both of these problems. Yeah.
Thank you, Samee bhai. আমরা তাহলে ইয়েটা রেকর্ডিংটা এখানেই শেষ করি। আপনার যদি সময় থাকে তাহলে কারো কোনো আর কথা থাকলে পরবর্তীতে করতে পারে। তো লেটস থ্যাঙ্কস সামি ভাই এগেইন, এবং সবাইকে নেক্সট সেমিনারে আসার ইনভাইটেশন থাকলো। থ্যাংক ইউ সামি ভাই।
এখানে কি আমরা এটা কন্টিনিউ